Hello, students. Hello, everyone. My name is Ghazala Firdaus, and I'm one of the biology faculty in Avanti Fellows. Okay, so we have completed AIET test, and we are going to solve the questions by understanding the answers and to understand briefly about the description and if there is an option correct and why that option is correct with the proper briefing and reasoning. Okay, so we're going to understand the biology part today. So this is biology class. Let's get prepared and start solving the questions. Okay, so we'll move ahead with the first question that says totipotent cell refers to. Okay, totipotent cell refers to an undifferentiated cells capable of developing into complete embryo, an undifferentiated cell capable of developing into an organ, an undifferentiated cell capable of developing into a system or an entire plant, cells which lack the capability of differentiating into an organ or system. So I'm going to give you 30 seconds. Okay, because you have already solved these questions, 30 seconds are more than enough. Just take your time and write down the answer. Okay, after 30 seconds, I'm going to solve it. Okay, okay. So we're going to understand now why, totipotent cell are worked here and which is the option. Clear? So what do you understand by totipotent cells, my students? See, totipotent cells are those cells which are undifferentiated. Okay, which are undifferentiated and they have a lot of capacity to develop into a complete embryo, or you can also say into a complete system. Okay, into a complete entire body or an entire system. Okay, you might be familiar with the tissue culture in biotechnology. What happened there? We will take a single this totipotent cell, and with the help of this single totipotent cell we are going to make the entire plant. Okay, so I hope that you are familiar with. Let's understand some of the points here. Totipotent cells can form all type of cells in a body, including the cells that are extraembryonic and placental. Okay, the only totipotent cells, or the embryonic cells inside the first couple of cells after fertilization. See, why this needs to be happening after fertilization? This part, or you can say the cleavage divisions where the blastomeres are formed, there the totipotency is in the next level. Why? Because from those blastomeres, the entire baby or the entire fetus has to be made, right? So that is why every individual there, every cell must be an totipotent cell. Okay, so totipotent cell have the capability of making an entire body. Okay, they have a possibility of making an entire body. So here, pluripotent cells can generate any one of the cell types that make up the body. Pluripotent are called as embryonic stem cells. Okay, so this is one of the points that I wanted to give you here. Now you can say which is the correct answer here. Okay, so an undifferentiated cells capable of developing into a complete embryo. Yes, that's true. Okay, so an undifferentiated cell capable of developing into organ. No, not just an organ. You can say in complete formation of embryo. Okay, an undifferentiated cell capable of developing into a system or entire plant body. Okay, system we cannot say, but plant body, yes. Okay, but still you have to choose which is more appropriate to you. Okay, which is more appropriate to you. The cells which lack the capability of differentiating, no, this is completely wrong. So you can choose the answer for this question is option A. Okay, and undifferentiated cells capable of developing into an complete embryo. As I have told you, after fertilization, they have to make an complete embryo. Okay, so all about this first question. Let's move ahead. Which of the following regulates the reproductive processes and associated behavioral expressions of organisms? Okay, I'm going to give you 30 seconds again here. Just look at the questions and write the correct, choose an option according to you. Okay, very easy question. All right. This is very easy, so I'm not going to to give you enough time. So, which of the following regulates the reproductive process and associate behavioral expressions of the organisms? See, we all know that in our human body, there is a need of hormone and the interaction with the environmental factors. Okay, so just hormones are not enough because we humans or living organisms, they need to interact with the living, environmental factors. Did you get my point? So here you can see hormones are responsible for the transitions between three phases, that is juvenile phase, reproductive phase, and senescence phase. We know that reproductive phase is adulthood, senescence is an oldhood, and juvenile is an younger, organism. So here what happens? These are by the hormones, but these effect of hormones are interacted with the environmental factors, which shows the behavior of an individual. Okay, responsible for regulating the reproductive process and also the associated behavioral expressions of organisms. Okay, so the temperament of an organism, or you can also say behavior of an organism. So everything depends not just based on hormones but also on environmental factor. Okay, you can take the organisms which are staying in a coastal area. They are adopted with the saline conditions, the hallow conditions. Okay, and the organisms which are present in the Western Ghats. Okay, so they are, used to having a very good humid climate with the proper availability of water. Okay, and some xerophytic plants, they do not need much water, but still they survive. They make sure to survive by storing the water into a gelatinous mucousy form. Okay, so here you can say plants also have hormone. Along with that, they need to interact with the environmental factor so that to show their behavioral expressions of any organisms, either animals or plants. Okay, so here you can say, so the answer for this question is D. Okay, abiotic components are nowhere included. So hormones are yes, but B is also yes. So we cannot take that as an individual. Why? Because there is always an interaction between hormones and environmental changes. Okay, so we are going to solve the next question here. Fibrous thickening of hygroscopic nature are found in which part of anther wall? Okay, so which part of the anther wall you are going to see the, fibrous thickening? Okay, this we have already studied and done. So I hope you remember the points which I have told you in the classes. Options are epidermis, endothelium, middle layers, and tapetum. Okay, please remember the structure of, anther. Anther TS section, if you remember, there we have talked about these layers, anther wall layers. Okay, so this is an easy question. Let's see the solution for this. So what happened in the anther, okay, in the section of an anther, in the middle of the epidermis, okay, in the middle of the epidermis are middle layers of an angiosperm anther sac wall. There is a single layer of radially elongated cells. We call them as endothecium. This is thecium, not thelium. Thecium is present. Okay, so these cells of endothelium are hygroscopic and they have fibrous thickening in their cell walls. Thus, they help in the dehiscence of mature anther. Okay, so anther takes time to mature, and after maturation of an anther, the pollen grains have to be dehisced out. So indirectly, these endothecium, they help in the dehiscence of the pollen grains out of the anther by their hygroscopic nature. Okay, as they have hygroscopic nature, they help in the expansion. Okay, or you can say what you can say contraction and expansion you can say. ओके तो क्या होता है बेटा, जब वहां पे मॉइस्चर कम होता है तो वो सिकुड़ने लगता है, ठीक है, जैसे वो कॉन्ट्रैक्ट होगा तो ओपन होंगे वॉल्स एंड डिहाइसेंस हो जाएगा, ठीक है. तो endothelium is the answer for this. So for those people who have forgotten where middle layer lies, where tapetum lies, where is epidermis, so for you, your homework is just go and look into the tiers of anther wall section. Okay. So here you can say answer for this question is B, endothelium. It was a very easy question. Okay. Now, so there's a next question on the screen. Following components are haploid, diploid, and triploid. Okay, so I have given some words here, okay, which are completely related with an, ovule. Okay, so ovule. So here you have to see the correct sequence of ascending order in the ploidy of ovule or seed. Okay, ovule or seed. Take some time. Take 1 minute of time, think, and then answer it. Okay. So I have given enough time. So let's see here. So what you have to do, you have to pick which is haploid, which is diploid, and which is triploid among this. So very best easy way, if I tell you, triploid you have seen only in endosperm after double fertilization. Okay, you have to look where is endosperm, which shows triploid condition in the options. Okay, so in the sequence, triploid is endosperm. Okay, endosperm is number 9. So 9 here, see, very easily you can recognize D. So here you can see which is diploid. Okay, diploid is, you can say, oospore, and the haploid is oosphere. Okay, so answer for this question is D, but still I will give you the solution and reasonings here. So what happened at the, in the ovule? Okay, we're talking about the structure of ovule here. In the structure of ovule at the micropylar end, so there are several number of archegonia cells are present. So what are archegonia cells? Are nothing but the female cells. Okay, each containing an oosphere. So oosphere is nothing but an egg. Oosphere represents to egg. So here what happens? This is before fertilization that we are talking. Okay, this is before fertilization. The pollen tube ultimately penetrates the neck of the archegonian. So after that, what happens? After that, there will be a process of fertilization. Okay, fertilization. So oosphere is nothing but egg. So this is before fertilization. So egg is haploid. We know that it is a gamete. Okay, so after fertilization, what happens? This oosphere will be converted into oospore. So oospore will become diploid now. So it is an already, oospore is an already fertilized female zygote that develops from fertilized oosphere in fungi and algae. Okay, so that you need to remember. And coming to the endosperm, see here I have told you. So endosperm is what? Endosperm is a triploid condition which helps in providing the nutrition. Okay, nutrition and enough amount of you can say moisture. Whatever the nutrition is needed, everything is provided there. Okay, so that you need to remember and also give some thought on this. Clear? So here, as you can see, option D is correct option. A, 1 is oosphere and 5 is oospore. 9 is endosperm. Okay, 9 is endosperm. Now, coming to the next question, the female parent plants produced unisexual flowers. Okay, so here you can see it's a very easy question. I will still give you some time. Take some time and then answer. Okay. So I have given you enough time now. We're going to solve this question now. The female parent plants produce unisexual flowers. Okay, that do not require emasculation, require emasculation are backed during birth stage, both A and C. So what do you mean by unisexual flowers? Unisexual flowers means these are the flowers which have only one sexual organ. Okay, that means unigender, you can say. Either female or male. Clear? So we are talking about the female now. Okay, so female plant which has been used in the process of hybridization. Here, in the unisexual flowers, there is no need of emasculation. Okay, so in this we are considering it as a female parent. Okay, so in this we are considering it as a female parent. Okay, so there is no need of emasculation as they possess either male flower or female flower. अगर दोनों ही होते तो हमको इमेस्कुलेशन करना था, हमको स्टेमन्स को रिमूव करना बहुत जरूरी था. बट यहाँ पे हम लोग क्या कर रहे हैं, यूनिसेक्सुअल फ्लावर्स है तो कोई जरूरत नहीं है हमको इमेस्कुलेशन करने की इन द फीमेल पेरेंट. ठीक है, तो क्योंकि होगा भी नहीं ना, फीमेल में एंथर्स होते हैं क्या, एंथर्स वुड नॉट बी देयर, सो देयर इज नो नीड ऑफ रिमूविंग और इमेस्कुलेटिंग. I hope that is clear to you. And then unisexual flowers are bagged during bud stage to prevent the contamination. Yeah, even though we are not doing emasculation, but we can be doing bagging. Why? To prevent the contamination. Say our female parent have stigma. It has the female reproductive organ, but we don't want it to be contaminated if we want pure lines. Okay, so in that case, what we are going to do, we are going to cover when the stigma of the bag flower attains the receptivity, मतलब वो जब mature हो जाता है, at that time, desired collected pollens are dusted on the stigma and then bagged. ठीक है, तो pollination करने के बाद भी bag कर सकते हैं और during the bud stage also we are going to bag. Why? To prevent the contamination. Okay, so bagging is done before the flower opening just to prevent the contamination by any chance. Okay, so here there is a note, emasculation and bagging methods of artificial hybridization to prevent interbreeding, while pollination is a process of inter and intra breeding. Okay, so I hope this is clear to you all. Okay, I know now you can see here what are the options. So female parent plants produce unisexual flowers. Here you can see A, option A, you can see do not require emasculation. Yeah, that's true. It's true. We do not require, emasculation here. Require emasculation? Definitely no. Are baggy during bud stage? Yes. So answer for this question is D, both A and C. Okay. Now, coconut water is the part of part of endosperm. Okay. So this is a direct asked question from NCRT, but still I can see many of you have made it mistake here. Okay. So it's a very direct question. So what are the options here? Cellular, free nuclear, cellular as well as nuclear, neither cellular nor free nuclear. Okay, I'm going to give you just 5 seconds. I'm giving you just 5 seconds. Take some time and then answer. Okay. So coconut water represents a nuclear, free nuclear endosperm. And the surrounding kernel represents the cellular endosperm. Because surrounding is malai. ओके या फिर नारियल जो नारियल क्या कहते हैं कोकोनट के साइड्स में होता है फ्री न्यूक्लियर एंडोस्पर्म इज द कोकोनट वाटर सो हियर व्हाट हैपेंस इन द फ्लूइड ऑफ कोकोनट वाटर देयर आर फ्री न्यूक्लियर एंडोस्पर्म लाइक दिस इट्स एन एंडोस्पर्म विथ फ्री न्यूक्लियाई okay so there are so many nuclei present in a system here okay so this is free nuclear endosperm surrounding kernel represents the cellular endosperm so as you can see so 121 का करेक्ट आंसर है बेटा फ्री न्यूक्लियर ठीक है तो इसका आंसर है फ्री न्यूक्लियर कोकोनट वाटर इज फ्री न्यूक्लियर एज बट द कर्नल जो मलाई होती है दैट इज सेलुलर बट हियर दे हैव आस्क्ड अबाउट द कोकोनट वाटर okay so this was again a direct question now coming to Chargaff's rule okay this question comes from molecular basis of inheritance so Chargaff's rule is applicable to single-stranded DNA and RNA, single-stranded RNA, double-stranded DNA, single-stranded DNA. Okay, so these are the options. Take some time and then try to solve the question. So I'm going to give you only 30 seconds because we have very less time and lot of questions to solve. Okay, we have to emphasis on solving more questions. Clear? ठीक है, Chargaff's rule. I hope you remember what is Chargaff's rule. Okay, let's understand here. Chargaff rule states that a DNA from any cell of any organism should have 1:1 ratio of pyrimidine and purine bases. More specifically, what we have to say, the amount of guanine should be equal to cytosine and the amount of adenine should be equal to thymine. This pattern is found in both the strands of DNA. As you know, DNA is a double-stranded DNA if we have to talk. So in that double-stranded DNA, what happens? So it looks somewhat like this. Okay, so wherever there is A, opposite would be T. Wherever there is C, opposite would be G. But you have to see A and T are binded with double bond, C and G are binded with triple bond. So here what happens, they have to be, anti-parallel polarity showing in the both the direction. Okay. And also if the amount of adenine is equal to thymine, then only you can properly form a double stranded DNA and make a DNA helix. If if you have to think, just imagine there is any number, deficit or an increased amount, then what happen, it will create the misnumber here. ठीक है, तो equal होना चाहिए बेटा, adenine should be equal to thymine and the guanine should be equal to cytosine. Only then a proper formation of double stranded DNA is formed. So this is Chargaff's rule. Okay, so single stranded RNA and DNA do not have complementary basis. So Chargaff's rule is not applicable to them. So that means what we are talking about, single stranded. Single stranded, देखो बेटा, single stranded में क्या होता है? There is no two strands, right? So there is no complementary basis, but it is only possible in the case of double stranded DNA. Okay, so here you can see the answer for this question is double stranded DNA, that is 130C. Okay, so this is Chargaff's rule. I hope this is clear. Okay, coming to next question. I will read out the question and I'll give you some time to solve this question. Select the incorrect statement regarding R strain of Streptococcus pneumoniae, which was used to prove the transforming principle. This should be italicized, but team has forgotten it to do it, so I'm going to underline it. Okay, yeah. So options are R strain bacteria has a mucus coat around it. It produced rough colonies. R strain is a non-virulent strain. Griffith used R strain in his transforming experiment. Okay, so take some time and then try to solve the question. Okay. All right. So I hope all of my students have done it very nicely because this has been completed in the classes. So I hope this is clear to everyone, but still I have to solve the question. Let's solve. So here, see, transforming principle is nothing but it is related to Griffith's experiment. Okay. So while Griffith's experiment had done, it had done to, understand the genetic material. It was done in the search of genetic material. Even though they did not recognize either it's in carbohydrate or protein or nucleic acid, RNA or DNA. इतना तो उनको पता नहीं चला, but एक simple चीज Griffith's experiment से conclude हुई थी, that is transforming principle. Let's understand what is that. So what Griffith has done, he took two strains. Okay, he took two strains. One is S strain, which is virulent. Okay, and there's another strain that is R strain. Okay, so S strain is virulent one. Virulent means it causes diseases. R strain means it is non-virulent. It is harmless. ठीक है, it is harmless. So virulent का another point है बेटा, इसमें virulence इसलिए है because it has a coat surrounding it. Okay, so coat is present over the bacteria. So here there is a no coat. Okay, no polysaccharide coat surrounding the R strain. So without coat, it is safer. R strain is safer, whereas the S strain is causing harmful, disease causing bacteria. Now what he done, he did the first step. He took the living S strain and injected into a mouse. So that F strain, which is living, it made the mouse die. Okay, and if you take out the cells of the mouse and you can see there is a presence of S strain, which is a virulent one or the disease causing bacteria. Okay, so he died with the pneumonia. Okay, so next step what he did, he took the living R strain, which is a non-virulent, and injected to a mouse. So there is no, sample of S strain found, but you can see mouse lives. Okay, because R strain is a non-harmful, it's a non-virulent. Why? Because it doesn't have an, coat surrounding it. Clear? So what happened in the third stage? So dead S strain is taken, but this dead is made, okay, or the death is made by heating up the S strain. S strain को कुछ time तक heat करने के बाद it leads to the death of S strain. Now, if we transfer that to one mice, then mice lives. Okay, so here mice lives. So there is no again scene of estuary. So in the fourth step, this is that transforming step which gave the transforming principle. So here what happened? Griffith took mixture of leaving R strain and dead S strain. Okay, leaving R strain and dead S strain. So here what should happen? Leaving R strain is safe, dead S strain is safe, but both together it made the death of mice. How is that happened? So this was observed by Griffith, and he concluded that something from the S strain is transferred to R strain, which causes the change of R strain into S strain. So in the mice you can see the S strain parts, some of the S strain remnants you can see. Okay, so this was transforming principle. A chemical substance from one cell is genetically transforming to another cell. Okay, so this was transforming principle given by Griffith. So let's see the option. Our strain bacteria has a mucus coat around it. Just tell me, does it have or does not have? It does not have, right? It was there in S strain, so that was creating the virulence in that S strain. But we have to choose an incorrect statement, so answer is A. But let's see other options. It produced rough colonies. Yes, it produced rough colonies. That means it's an correct statement, not incorrect. R strain is non-virulent strain. Yes, incorrect statement. तो नहीं है तो इसीलिए wrong. Griffith used R strain in this transforming experiment. No, he used normal R strain. There he saw the change of R strain into S strain. Okay, so answer for this question is simply A. Okay, so we are going to solve the next question now. The haploid content of human DNA contains. Options are given. So please look into the options and then try to solve the question. Okay, this is the direct NCERT question. It's an easier question. But, confusion arises here. Take some time, think, and then we are going to answer it. Okay. So haploid content means what? Here we are talking about only half of the human DNA. Okay, तो total अगर human DNA मिला है तो that would be diploid content. ठीक है, तो यहाँ पे क्या हो रहा है 3.3 into 10 to the power of 9 base pairs. So this is the haploid content of human DNA. It's a direct NCERT question. इसमें कोई doubt ही नहीं है और होना भी नहीं चाहिए. So haploid content of human DNA is called as human genome. Okay, that is 3.3 10 to the power of 9 base pairs. Whereas diploid content is 6.6 into 10 to the power of 9 base pairs. Okay, it's a direct question taken from the NCERT statements. Okay, now there comes another question from the principle of inheritance and variation. Law of independent assortment is always true or applicable if two genes are located on same homologous chromosome, different chromosome, same locus of different chromosome, different locus of same chromosome. Okay, think, and then choose your option. Okay. So, as you know, Mendel has given three laws: law of dominance, law of segregation, and the law of independent assortment. So, law of independent assortment is only seen in the dihybrid cross or where we have taken the two pair of contrasting triads. Okay, so दो characters को लिया है. So, monohybrid cross was done by only one trait. Okay, either height of the stem or either color of the pod or color of the say a flower or the shape of the pod, कुछ भी हो, ठीक है. बट यहाँ पे हम लोगों ने दो पेड़ लिए हैं बेटा. ओके, दैट इज शेप ऑफ सीड. ओके, शेप ऑफ सीड and color of seed. Okay, color of seed, this we have taken. So here what happened here in this, r is round, small r is wrinkled, capital Y is yellow, small y is green. Okay, so यहाँ पे क्या हो रहा है बेटा, here what happens in the law of independent assortment. Okay, so here what happened, you can see the statement here. Mendel's law of independent assortment states that allele of different genes assort independently of one another during the gamete formation. ठीक है, तो अब यहाँ पे देख लो बेटा, इसका नेक्स्ट प्रोसेस क्या है. यहाँ पे भी गैमीट फॉर्मेशन करना है ना. तो गैमीट फॉर्मेशन के टाइम पे, ड्यूरिंग द गैमीट फॉर्मेशन, दिस कैपिटल आर कैन इंडिपेंडेंटली फ्यूज विद कैपिटल वाई, or it can also independently fuse with small y. So this is independent assortment. Assort means joining. Independent means freely, without anybody's stress or force. ठीक है, तो कैपिटल Y, कैपिटल आर कैपिटल वाई के साथ भी जा सकता है, कैपिटल आर स्मॉल वाई के साथ भी जा सकता है. So this is how there is an independent assortment. So small r can fuse with either capital Y or either small y. ठीक है, so like this there is an independence in choosing a opposite allele for making a gamete. ठीक है, तो gamete formation में there is an total independence during the gamete formation to assort among the alleles or chromosomes. ठीक है, see when we are talking about factor or gene, आपको पता है ना, what Mendel had told, he had talked about factor which is now called it as a gene. But where is gene present? Gene is present inside a chromosome. ठीक है, अब ये क्रोमोसोम है, तो this is gene and this gene is present on a specific locus. Okay, so this gene might have multiple alleles or you can say forms of allele. एक से ज्यादा forms हो सकते हैं allele के. ठीक है, तो this is gene and this gene has forms. Okay, of gene is allele. Okay, so if you have to take the height of the stem, there are two. Okay, one is for tall and another one is for the dwarf. Okay, so T allele and the small t allele. ठीक है, तो small t is the indication of recessiveness, capital is indication of tallness. ठीक है, so like this every gene has alleles. ठीक है, every gene has alleles and every chromosome has genes and these genes have the capacity for showing the phenotypic expressions. ठीक है, तो independent assortment is done during the gamete formation and it must have two pair of contrasting characters. Only then it can independently assort with a different chromosome. ठीक है, तो different chromosome. तो मैंने यहाँ पे देखो ऐसे दिखाया है अलील के टाइप में. अगर मैं यहाँ पे क्रोमोसोम ले लूं तो 4 क्रोमोसोम आ जाएंगे, है ना. सो नाउ दिस इज फ्री टू ज्वाइन विद दिस एंड विद दिस. ठीक है, तो ऐसे होता है. क्लियर, तो यहाँ पे अभी ऑप्शंस देखते हैं. Law of independent assortment is always true and applicable if two genes are located on same homologous chromosome. No, देखो ये दोनों तो एक ग्रुप है, ये दोनों तो एक ग्रुप है, so there are two homologous chromosomes here. So no, option A is incorrect. Different chromosome, yeah, there is a different chromosome, it might be correct, but let's see the other option. Same locus of different chromosome need not to be necessary. ये जरूरी नहीं है बेटा, दिस इज नॉट नेसेसरी, नीड नॉट टू बी नेसेसरी. ओके, सो डिफरेंट लोकस ऑफ सेम क्रोमोसोम डेफिनेटली इनकरेक्ट. क्यों? बिकॉज देयर इज अ नीड ऑफ अनदर अलील, अनदर फैक्टर, अनदर क्रोमोसोम. सेम क्रोमोसोम में कोई खिचड़ी नहीं पक सकती. सो आंसर फॉर दिस क्वेश्चन इज डिफरेंट क्रोमोसोम. ठीक है, क्रोमोसोम से दूसरा होगा एंड उसके साथ ज्वाइनिंग भी दूसरे क्रोमोसोम के साथ होगी ड्यूरिंग द गैमीट फॉर्मेशन. व्हाई? बिकॉज हियर वी हैव टेकन अ पेयर ऑफ कॉन्ट्रास्टिंग कैरेक्टर्स, दैट इज डाइहाइब्रिड. Okay, so I hope this is clear and understood to everyone. Now, coming to the next question, how many phenotypes are produced from the test cross of this. Take some time, solve it, and then we are going to answer it. Okay, so I think I have given enough time. So, how many phenotypes? What is the understanding of phenotype? Phenotype are those which gives the expression on physical side. Okay, physical expressions. Genotype क्या होता है? उसका genetic makeup होता है. ठीक है, that internal genetic makeup is genotype. Phenotype is the the physical expressions that we can easily see through our eyes is phenotype. Now we have to do the test cross of this. ठीक है, तो ये आपके क्वेश्चन में दिया है, तो इसका टेस्ट क्रॉस करना है. So for doing test cross, what is test cross? Test cross is simply a cross between an F1 hybrid with a recessive parent. ठीक है, it is done with the recessive parent. This is test cross. So in test cross, an individual of an unknown genotype, okay, genotype must be not known, ठीक है, is crossed with the homozygous recessive individual to identify the form or proportion of offspring phenotypes. ठीक है, तो हम लोगों को नहीं पता है कि इसके जीनोटाइप क्या है, तो जीनोटाइप को पता लगाने के लिए हम लोग टेस्ट क्रॉस करते हैं बाय क्रॉसिंग द हाइब्रिड विद द रिसेसिव पेरेंट. ठीक है, तो अब यहाँ पे हम लोग क्या करेंगे, टेस्ट क्रॉस कर रहे हैं हम इसका इसके रेसेसिव पेरेंट के साथ एज वी हैव शोन इन द फिगर. ठीक है, यहाँ पे फिगर ले लिया है. तो यहाँ पे इसको क्रॉस करने से पहले व्हाट वी आर गोइंग टू डू, वी आर गोइंग टू टेक आउट द गैमीट्स. तो देखो एक टाई हाइब्रिड क्रॉस के लिए इतने गैमीट्स निकले हैं. ठीक है, सो 1 2 3 4 gametes. So there are 4 gametes, and by doing the Punnett square, if you plot the Punnett square or if you simply cross, okay, so what you are going to get in the F1 generation, these are the 4 possible different phenotypes you're going to see in the F1. Okay, so it can either be F1 or you can say any unknown genotype. Okay, so unknown जीनोटाइप कुछ भी हो सकता है यहाँ पे. तो यहाँ पे आप देख लो, therefore 4 different phenotypes are produced in F1 as a result of this test cross. So you got your answer. How many phenotypes are produced from this test cross? That is 4 phenotypes. Option C is the correct option. Okay, so we'll see the next question now. So gynecomastia is associated with which disorder? Options are Down syndrome, color blindness, Turner syndrome, Klinefelter syndrome. Think, think, and then answer. Very easy question if you remember concepts. If you don't remember concepts, then I can't do anything. Okay. So what is gynecomastia? Gynecomastia is nothing but a symptom of enlargement of breast. Okay, so enlargement of breast is nothing but gynecomastia. Okay, so here there are 4 options: Down syndrome, color blindness, Turner syndrome, and Klinefelter syndrome. So to give the answer, you must be knowing which are the disease, what there is, its function. ठीक है, तो पहले यहाँ पे देख लेते हैं बेटा, what is Down syndrome? So here I can say Down syndrome is the trisomy. Okay, so Down syndrome is trisomy of 21st क्रोमोसोम. ठीक है, तो 21st क्रोमोसोम में बेटा वहां पे क्या होता है, ट्राइसोमी. जैसे ए हो गया, एक मिनट, ये ए नहीं लेती हूँ मैं, नंबर ले लेती हूँ, आपको समझने में इजी हो जाएगा। यहाँ पे नंबर ले लेंगे हम लोग। ओके, सो हियर वी कैन टेक, सो 44 होना चाहिए, है ना, 44 होके इधर एक्स एक्स और एक्स वाई होना है, ये नॉर्मल होता है। 44 और ऑटोसोम्स एक्स एक्स और एक्स वाई इज अलोसोम, एक्स एक्स इन द केस ऑफ फीमेल, एक्स वाई इन द केस ऑफ मेल। तो यहाँ पे क्या होता है, 44 प्लस 1 हो जाता है। सो ये जो प्लस 1 है ये कहलाता है ट्राइसोमी। ओके, सो दिस इज ट्राइसोमी, दैट इज 2n plus 1. So this is trisomy. This is seen in the 21st chromosome, which is seen by the Downs syndrome. ओके, यहाँ पे सिम्टम्स होते हैं जैसे मेंटल रिटार्डेशन, थोड़ा फिजिकल कुछ प्रॉब्लम हो जाना, तो ये सब चीजें। देन कमिंग टू द टर्नर सिंड्रोम। ओके, आई विल टेल यू टर्नर सिंड्रोम। टर्नर सिंड्रोम इज सीन यूजुअली इन द फीमेल्स। तो 44 क्रोमोसोम्स तो होते हैं, तो लड़कियों में एक एक्स क्रोमोसोम एब्सेंट हो जाता है। Okay, one X chromosome is absent. So this leads to the deficiency or many problems in the females. ठीक है, तो complete development of ovaries is not possible and the proper development of breast is not possible. तो एक complete female में development ही नहीं होता है. This is Turner syndrome. Okay, what is Klinefelter syndrome? तो यहाँ पे क्या होता है बेटा, this is seen in males. ये होता है females में, ठीक है, बट लिनेसिल्टर सिंड्रोम इज सीन इन मेल। तो यहाँ पे क्या होता है, एक्स वाई क्रोमोसोम के साथ एक एक्स्ट्रा एक्स आ जाता है। सो दिस इज सीन इन मेल, ठीक है। तो लीने फिल्टर्स में आप देखोगे देर इज एन एनलार्जमेंट ऑफ ब्रेस्ट, ठीक है। ही इज मेल बट ही ऑल्सो हैज द कैरेक्टर्स ऑफ फेमिनाइन, फेमिनाइन कैरेक्टर्स भी इनके अंदर होते हैं, ठीक है। सो दिस इज लीने फिल्टर सिंड्रोम। So here, enlargement of breast is nothing but gynecomastia, which is seen in the Klinefelter syndrome. Okay, here you can see. So this is a person who has a less body hair, okay, as just in the case of females, and there is a breast enlargement with an extra X chromosome. XY should be there, but an extra X chromosome, which makes the problem in male. A genetic condition in which a male is born with an extra copy of X chromosome. This is Klinefelter's syndrome. Okay. So this is very clear and simple question. Coming to next question. A boy has a normal brother and a colorblind sister. What is the true about his parents? So you have to plot the cross here. आपको यहाँ पे cross बनाना है so that you get the normal brother and the colorblind sister to know their parents. ठीक है, तो parent कैसे थे जानने के लिए आपको यहाँ पे बनाना पड़ेगा एक क्रॉस, ठीक है। सो लुक एट द ऑप्शंस वंस एंड देन ट्राई टू मेक द क्रॉस। आई विल गिव यू अ 1 minute हियर बिकॉज इट नीड्स सम टाइम टू डू अ क्रॉस। ओके, क्रॉस करने के लिए 1 minute टाइम लगेगा, तो 1 minute आपको मैं दूंगी। जस्ट टेक सम टाइम एंड डू इट। I know many of my students will be able to do it because we have done these kind of questions in the classes. कि क्लासेस में हमने ऐसे क्वेश्चंस किए हैं बेटा, तो कोई मुश्किल नहीं होगी आपको सॉल्व करने के लिए ये क्वेश्चंस। ओके, so we are going to check now. All right. So here you can see if you cross a normal mother with a colorblind father, ठीक है, तो आपको क्या मिलेगा बेटा, ये है अफेक्टेड फीमेल। affected female, here female carrier, affected male, normal male. So what do you get here? You get two affected people, female and male. One female is carrier and another male is affected. So what they say here, they say a boy has normal brother and the color blind sister. ओके, तो कौन है यहाँ पे, अब देख लो, नॉर्मल ब्रदर भी है और कलर ब्लाइंड सिस्टर भी है. सो दिस क्रॉस इज obtained बाय डूइंग द क्रॉस ऑफ नॉर्मल मदर एंड कलर ब्लाइंड फादर. ठीक है, तो यहाँ पे आप देख लो, ऑप्शन बी वुड बी द करेक्ट ऑप्शन. हिज फादर वॉज कलर ब्लाइंड बट द मदर वॉज नॉर्मल. ठीक है, तो आप ऐसे ही हर चीजों का करते जाओ. ठीक है, इफ यू हैव टू डू इट कंटिन्यूअसली सो दैट टू चेक विच अमॉन्ग द क्रॉस वुड बी करेक्ट वन. अभी मेरे पास टाइम नहीं है, वरना मैं दिखा देती आपको. ठीक है, जैसे यहाँ पे ले लो, अब बोथ फादर एंड मदर वर नॉर्मल. ठीक है, तो मदर नॉर्मल है, फादर नॉर्मल है, तो यहाँ पे कोई बात ही नहीं है. सो एवरीथिंग इज नॉर्मल हियर. ओके, नाउ कंसिडर बोथ फादर एंड मदर आर कलर ब्लाइंड. ठीक है, दोनों ही फादर एंड मदर आर कलर ब्लाइंड. ओके, सो कलर ब्लाइंड होंगे तो व्हाट डू यू गेट हियर? देखो, सारे ही कलर ब्लाइंड निकलेंगे. सारे कलर ब्लाइंड है. सो दिस ऑप्शन इज ऑल्सो नॉट करेक्ट, दिस ऑप्शन इज ऑल्सो करेक्ट. अब ये भी करो तो आपको ये भी यहाँ पे भी मिल जाएगा. ठीक है, तो अब देख लो, हिज फादर इज नॉर्मल, वो भी कर देते हैं, क्यों आपको मतलब दिल दुखे. ओके, मदर इज कलर ब्लाइंड. ठीक है, मदर इज कलर ब्लाइंड हियर. सो इफ यू क्रॉस हियर, okay, both the males are affected, there is no normal son. Okay, both are carriers, carrier sisters. Okay, so question में क्या पूछा है? Normal brother and colorblind sister, both the sisters are carrier and both are affected males. So hence answer for this question is the B. Okay, so this is very simple question, just you have to take some time and do it. Okay, so the next question is bar body is associated with. ठीक है, तो बार बॉडी बेटा किन चीजों से एसोसिएट होती है? ओके, ऑप्शंस आर सेक्स क्रोमोसोम ऑफ फीमेल, sex chromosome of male, autosome of female, and autosome of male. Okay, I'll give you 30 seconds to choose your option. Please remember bar bodies are very important. Okay, they have been asked in previous questions. ठीक है, तो इसके बारे में थोड़ा इंफॉर्मेशन आप जान लीजिए. Okay, we'll look into the solution now. So here what's there? It is the bar body is formed due to inactivation of genes on one of the X chromosome present in the female. तो ये फीमेल से एसोसिएटेड है, दैट टू एक्स क्रोमोसोम. ठीक है, इतना देखते ही आपको आंसर पता चल जाना चाहिए, हाँ भाई सेक्स क्रोमोसोम फीमेल, ये इज करेक्ट. बट और ज्यादा आपको इन्फॉर्मेशन चाहिए. दिस इज करेक्ट, ए इज करेक्ट. ठीक है, but just by looking at the first line you can tell the option, but still you need some more explanation on this. The bar body is formed due to inactivation of genes on the X chromosome present in females. Why it is done? It is done to prevent the double expression of X chromosomes. ठीक है, डबल एक्सप्रेशन नहीं होना चाहिए एक्स क्रोमोसोम का, तो इसको प्रिवेंट करने के लिए बार बॉडी इज फॉर्म ड्यू टू द इनएक्टिवेशन ऑफ सम जीन्स ऑन एक्स क्रोमोसोम, वन ऑफ द एक्स क्रोमोसोम, नॉट बोथ. ठीक है, देखो बेटा, फीमेल्स के पास तो दो एक्स क्रोमोसोम है ना, तो हमको एक ही चीज दो दो बार नहीं चाहिए, बस एक चीज ही काफी है. तो एक इनएक्टिव हो जाती है. ठीक है, वन ऑफ द एक्स जीन विल गेट inactive. Okay, so compared to male who have only one X chromosome, so and zero bar body, females have two X chromosome and one bar body. ठीक है, तो मेल्स के पास देखो बेटा, इन द केस ऑफ मेल देयर इज ओनली वन एक्स क्रोमोसोम, तो वन एक्स को तो एक्सप्रेस होना ही है, सो देयर इज नो सेकेंड कॉपी ऑफ एक्स, तो यहाँ पे देयर इज नो बार बॉडी, बार बॉडी आपको नहीं मिलेगी. इन द केस ऑफ फीमेल यहाँ पे दो एक्स क्रोमोसोम से रिपीट हुई हैं, तो इन केस में आपको एक एक्टिव होगी और एक आपको बनेगी बार बॉडी. ठीक है, सो दिस यू नीड टू remember, it's a random process. ये होता है, ये common है. It's a random process which occurs in the case of females, which is seen in the sex chromosome of X. ठीक है? तो आंसर फॉर दिस क्वेश्चन इज ए. ऑटोसोम तो बिल्कुल नहीं, सेक्स क्रोमोसोम ऑफ मेल भी नहीं. ठीक है? नाउ कमिंग टू द नेक्स्ट क्वेश्चन, द प्राइमरी oocytes आर इन विच स्टेज ऑफ सेल डिविजन? सो आई होप यू रिमेम्बर oogenesis. अगर oogenesis याद है तो उसमें आप इन स्टेजेस को देखे होंगे. याद करो बेटा. आई विल गिव यू सम 40 seconds. Remember and then try to solve the question. It's a very easy question. Okay. So let's see here in the diagram, this is oogenesis. Okay, oogenesis. So the process of oogenesis had already started when a baby girl or the child has still in the mother's womb. ठीक है, जब वो अभी भी मदर के वूम में है तब से ही उसका क्या होता है बेटा, प्रोसेस oogenesis का स्टार्ट हो जाता है, अ फीमेल बॉडी. ओके, सो oogoниयम इज द फीमेल मदर सेल. ओके, so female mother cell है. Oogonia will undergo mitosis, which is an equational division, to give the diploid primary oocyte. So all this is happening before the birth of a girl child. ठीक है? Now this primary oocyte will be arrested in the primary oocyte stage of the prophase 1. तो अभी क्या हो रहा है बेटा समझो. Oogonia is undergoing mitosis. Okay, it is undergoing mitosis and it has started meiosis. माइटोसिस को कंप्लीट करके उसने मायोसिस स्टार्ट किया है, बट स्टार्ट करते ही प्रोफेज 1 में अरेस्ट हो गया. इट हैज बीन अरेस्टेड देयर. ठीक है, तो अब क्या हो गया, बेबी का बर्थ हो गया एंड आफ्टर द बर्थ दैट यंग फेज ऑफ द फीमेल चाइल्ड, तो उस टाइम पे कुछ चेंजेस नहीं होंगे. बट एज सुन एज द फीमेल अटेन द प्यूबर्टी, तो ये जो हॉल्ट हुआ था, ठीक है, जो oogenesis प्राइमरी oocyte में इन द प्रोफेज 1 स्टेज में हॉल्ट हुआ था, that will resume and the meiosis continues. So here what happened, primary oocyte will undergo meiosis 1 to give the secondary oocyte and polar body. Okay, so this secondary oocyte will be arrested in the metaphase 2 of meiosis. ठीक है, अगेन ये meiosis 2 में halt होगा. ठीक है, तो अब क्या होगा, what happens, so if okay, if a sperm is available for this secondary oocyte, अगर sperm मिला है secondary oocyte को, तो उस टाइम पे ovulation होगा, then meiosis complete होगा following fertilization to give the fertilized egg, that is zygote. ठीक है, तो when this is possible, when there is an availability of sperm. अगर sperm नहीं है तो उस टाइम पे this secondary oocyte will remain as it is and it will be flushed out in the menstrual cycle. ठीक है, तो ये पूरा प्रोसेस है oogenesis का. Okay, so here you can see 154 question. So primary oocytes are in which stage of cell division? They are in the prophase 1 of meiotic division. एक और एक सिंपल फंडा मैं बताऊं, माइटोसिस में तो ये फेस ही नहीं होते, मतलब ये 1-2 की बात ही नहीं होती. ये 1-2 स्टेज 1-2 एंड इज सीन इन द मायोसिस. ठीक है, सिर्फ मायोसिस में स्टेज 1 मायोसिस, स्टेज 2 मायोसिस इज सीन. माइटोसिस में इक्वेशनल डिविजन होता है, ओनली 4 फेजेस, प्रोफेज, मेटाफेज, एनाफेज एंड टेलोफेज. whereas meiosis में सेम फेजेस 2 स्टेजेस में होते हैं. ओके, 1-1, 2-2, लाइक दिस. तो यहाँ पे बहुत सिंपल आंसर था ये. ठीक है, सो दिस इज क्लियर आई गेस. नाउ कमिंग टू द नेक्स्ट क्वेश्चन, find the incorrect match with respect to increase in the levels of following hormones: oxytocin, prolactin, progesterone, luteinizing hormone. Okay, so I'm going to give you exactly 60 seconds to choose your option. Right. So I have given you enough time now. So what we are going to do, we are going to solve this question now. तो यहाँ पे देख लो बेटा, you have to find the incorrect match. आप करते ये हो, you in the urgency of solving the questions, you do not read the question properly. They have asked to match the incorrect one. What you do, you see the correct one and you immediately tick it out. You have to see all the options and pick the odd one. Okay, with respect to increase in the levels of following hormones. Okay, so what does the oxytocin do? Oxytocin helps in the uterine contraction during the labor. But the oxytocin is one such hormone which helps during the process of parturition. Okay, when the baby is trying or the fetus is trying to come out of the womb, at that time uterus have to contract and the cervix has to be relaxed. So that cervix ka relaxation is done when hormone relaxin, whereas oxytocin is helped in the contraction of uterus so that we should push the baby out of the uterus. Okay, so correct option oxytocin with contraction during labor, ये correct है, but हमको choose करना है incorrect. Prolactin, lactation after childbirth, definitely true. See, when a lady has gotten pregnant, so at that time what happens? Many hormones start to originate. At that time, even prolactin is there, which has started the synthesis of making milk or formation of milk. ठीक है, तो 3 या 4 months के बाद फॉर्मेशन ऑफ मिल्क स्टार्ट हो जाता है इन द ब्रेस्ट ऑफ अ प्रेग्नेंट वुमन, सो दैट इट इज द जॉब ऑफ फैसिलिटेटिंग द लैक्टेशन आफ्टर द चाइल्ड बर्थ. जब बच्चा पैदा हो जाएगा उसके बाद लैक्टेशन करवाना होता है, तो उसके लिए प्रोलैक्टिन हार्मोन इज रियली नेसेसरी फॉर द लैक्टेशन आफ्टर चाइल्ड बर्थ. सो दिस इज ऑल्सो करेक्ट, प्रोलैक्टिन. Progesterone, uterine contraction, no. Progesterone doesn't do the uterine contraction. Progesterone helps in the maintaining. Okay, so progesterone helps in the formation of endometrium lining. ठीक है, तो endometrium को thick बनाते जाएगा for the safe, for the safeguarding and providing an safe environment for the formation of fetus. ठीक है, तो फीटस फॉर्म होगा तो इम्प्लांटेशन होते होते ये सब होता है. तो प्रोजेस्ट्रॉन का होना बहुत जरूरी है क्योंकि एंडोमेट्रियम में फॉर्मेशन ऑफ मेनी लेयर्स, थिक फॉर्मिंग ऑफ यूटेराइन इज डन बाय द प्रोजेस्ट्रॉन. ठीक है, आपको पता है ओव्यूलेशन के बाद जो कॉर्पस ल्यूटियम बन जाता है, दैट कॉर्पस ल्यूटियम इज गोइंग टू सेक्रेट दिस प्रोजेस्ट्रॉन. ठीक है, so that corpus luteum will remain as it is after fertilization to continuously keep giving out the progesterone. If fertilization has not happened, then corpus luteum would get degenerated, okay, which will not eventually lead to give this much number of progesterone. जैसे प्रेगनेंसी में नहीं, नॉर्मल केस में मैं बता रही. ठीक है, now coming to luteinizing hormone. So luteinizing hormone, you can see here, this is called as okay menstrual cycle का chart. Okay, so here we will see LH is a luteinizing hormone. आपको सबको पता है during ovulation there is a surge of LH to give out the secondary oocyte from the mature Graafian follicle, है ना. तो यहाँ पे एकदम से LH increase होता है to facilitate the ovulation, to stimulates ovulation. Even this is correct. The incorrect option is C, which makes the correct option for this question, progesterone uterine contraction. Okay, easy था. अगर आपने हार्मोन एक्शन पढ़ा है तो आपको ये सॉल्व करना बिल्कुल इजी हो जाएगा. ठीक है, now coming to the next question. Which of the following is the correct statement? Okay, parturition is induced by complex neuroendocrine mechanism. The signals for parturition originate from the fully developed fetus and the placenta, which induces mild uterine contraction called fetal ejection reflex. Oxytocin hormone helps in contraction of uterine muscles. Relaxin hormone helps in dilation of pubic symphysis. Okay, I'm going to give you 1 minute here. If you had read NCRT, you would be easily solving this question because sentences are really directly taken from NCRT. Okay, so I will give you 60 seconds, think, and then answer which is the correct one. Choose your correct option. ओके, आई हैव गिवन यू एनफ टाइम, इसके लिए इतना टाइम काफी है. नाउ पार्चुरेशन इज व्हाट? पार्चुरेशन इज द प्रोसेस ऑफ इजेक्टिंग द बेबी आउट ऑफ द वूम्ब और द यूट्रस. ठीक है, यूट्रस से बाहर इजेक्ट करने वाले प्रोसेस को बोलते हैं हम लोग पार्चुरेशन, और नॉर्मल वर्ड्स में डिलीवरी ऑफ द बेबी. ओके, सो इट्स एन इंड्यूस्ड कॉम्प्लेक्स न्यूरो एंडोक्राइन मैकेनिज्म. ये इंड्यूस होता है, नेचुरल नहीं है. इंड्यूस होता है विद द हेल्प ऑफ न्यूरो एंडोक्राइन मैकेनिज्म. तो कौन इंड्यूस कर रहा है? इंड्यूस, ओके, इंडक्शन इज डन बाय द फुल्ली डेवलप्ड fetus and placenta. ठीक है, so here what happened in the process of parturition, signals are originated from fully developed fetus and placenta. जो fetus fully develop होता है वो induce करता है signals को, and these signals will give the signals to the brain. ठीक है, वो ब्रेन को सिग्नल देगा कि हाँ भाई मैं ग्रो हो गया हूँ, आई हैव टू कम आउट ऑफ द वूम नाउ. ओके, सो दिस विल लीड टू द माइल्ड यूट्राइन कॉन्ट्रैक्शन. नाउ व्हाट ब्रेन विल डू? ब्रेन और यू कैन से हाइपोथैलेमस. ओके, तो सिग्नल्स जाएंगे हाइपोथैलेमस को. हाइपोथैलेमस विल मेक श्योर टू सेक्रेट द ऑक्सीटोसिन. सो दोज ऑक्सीटोसिन हेल्प इन द यूट्राइन कॉन्ट्रैक्शन. ठीक है, जैसे यूट्राइन कॉन्ट्रैक्शन होगा देयर इज एन reflux. What is that reflux? Fetal ejection reflex. Okay, so as you can see, contraction of uterus is happening, the fetus will be ejected out. Okay, so oxytocin acts on uterine muscles, leading to stronger and stronger uterine contractions. During the entire process of labor, oxytocin का बहुत important role है. कई फीमेल्स को ऑक्सीटोसिन एनफ अमाउंट में प्रोड्यूस नहीं होता. उस केस में there has been an artificial injections given to the female to do the normal delivery by giving these injections of oxytocin dosage. Okay, so this further continues to stronger and stronger contraction. As a result, the baby is expelled out of the uterus with placenta through the birth canal. Okay, so you have understood the concepts now. Come, let's see which are the correct statements. Parturition is induced by complex neuroendocrine mechanism. Yes, this is correct statement. Signals for parturition originate from fully developed fetus and the placenta, which induces the mild contraction called, yeah, this is also correct. Oxytocin hormone helps in the contraction of uterine muscles. Yes, correct. Relaxin hormone helps in the dilation of pubic symphysis and cervix. Okay, so both cervix and pubic symphysis. Pubic symphysis nothing but the bone, the pubic bone beta, वो थोड़ा ऐसे broad हो जाता है, so that the baby's head has to come out and the eventually complete ejection of baby. So here we can see all the options are correct. So option D is the correct answer here. Okay, so now coming to this question. If a person is suffering from severe male infertility due to a very few sperm, oligozoospermia, or even no live sperm, which is the term given azoospermia in the ejaculate, it can overcome by which process? This should offer couples an alternative to using donor sperm. Okay, so this should offer couples an alternative to using donor sperm. ये sentence को ध्यान से पढ़ो. If this sentence had not given, then answer would have been completely different. Because this sentence is present, you have to choose the option now. Okay, chalo. I'm going to give you only 40 seconds. Take your time and then solve the question. Okay, so I have given you enough time. तो यहाँ पे क्या बता रहा है? Male person is suffering from severe infertility, both like azoospermia and also azoospermia, दोनों ही chances हैं. तो इस केस में क्या हो सकता था बेटा? अगर ये स्टेटमेंट नहीं होता तो इन दिस कंडीशन ओनली इतना ही क्वेश्चन होता, तो इसका आंसर होता आर्टिफिशियल इंसेमिनेशन. ओके, सो आर्टिफिशियल इंसेमिनेशन. So here what happens in this process, a donor sperms are inseminated either in the vagina or in the uterus. ठीक है, तो donor sperms को हम लोग कर सकते थे, but यहाँ पे क्या दिया है? This should offer couples an alternative using to donor sperm. ठीक है, तो इस कपल को मतलब इस टाइप के पर्सन को इसको क्या करना है, डोनर को यूज नहीं करना है, फिर भी इसको अपना ही जीन्स रखना है। तो इस टाइम पे हम लोग क्या करेंगे, वी विल चूज एटलीस्ट वन गुड स्पर्म। तो उस एक स्पर्म अगर है तो भी उसके साथ हम प्रोसेस करेंगे, दैट प्रोसेस इज कॉल्ड एज इक्सी। तो इंट्रासाइटोप्लाज्मिक स्पर्म इंजेक्शन। So with that one single sperm, what we are going to do, we are not going it letting waste। ठीक है, इसको ऐसा ऐसे लेट गो नहीं कर रहे हैं, इसका वेस्ट नहीं कर रहे हैं हम लोग। व्हाट वी आर गोइंग टू डू, वो एक स्पर्म को हम डायरेक्टली क्या कर रहे हैं, इंजेक्ट कर रहे हैं इनसाइड द एग। ठीक है, तो यहाँ पे देख लो है हमारा स्पर्म। ओके, लैबोरेटरी प्रोसीजर इन विच अ सिंगल स्पर्म ऑफ मेल पार्टनर इज इंजेक्टेड डायरेक्टली इनटू एन एग ऑफ फीमेल पार्टनर। ठीक है, तो अब हमने डायरेक्ट प्रोसेस कर दिया, अब क्या होगा, फर्टिलाइजेशन होगा। आफ्टर फर्टिलाइजेशन द एम्ब्रियो इज इम्प्लांटेड इन द वुमेन्स यूट्रस। ठीक है, तो ये होता है इक्से, intracytoplasmic sperm injection, and this is done when the person doesn't want to use the donor. So alternative of using donor is using their own sperm, but at least one sperm is healthy and enough, we have to pick that one and do the ICSI out of it. Okay, so this is the answer for this question. Now, ploidy of spermatogonia, primary spermatocyte, secondary spermatocyte, and spermatid is respectively. Okay, so these are the options. And these are your options. Okay, so this is easy question बेटा, आपको इतना डिफिकल्टी नहीं होनी चाहिए. So this is spermatogenesis, okay, formation of sperms. So here there is a spermatogonia, male mother cells. Okay, so here it will undergo mitosis to give the diploid primary spermatocyte. Now occurs meiosis 1, which give haploid secondary spermatocyte, which undergo meiosis 2 to give spermatids, which will eventually undergo spermiogenesis to give the spermatozoa. This is the spermatogenesis. अभी यहाँ पे देख लो बेटा प्लॉइडी। ठीक है, प्लॉइडी यहाँ पे आपको देखना है। Spermatogonia is diploid. Primary spermatocyte is diploid. Secondary spermatocyte is haploid. Spermatid is haploid. So which is the correct answer for this? Spermatogonia, primary spermatocyte. हाँ, तो अब यहाँ पे देख लो, ये ऑप्शन तो बिल्कुल रॉन्ग है, ये भी रॉन्ग हो गया. So this is correct one. Okay, this one is correct one. Now, coming to the last question of this session. Okay, match the items given in column 1 with column 2 and select the correct option given below. Okay, you have to choose the correct option here. I will give you 40 seconds to answer this question. Okay, so ultrafiltration कहाँ पे हो रहा है बेटा? This is happening in the glomerulus or also called as Malpighian corpuscle. ठीक है, जो कॉन्सेंट्रेशन ऑफ यूरिन होगा वो होगा हेनलेस लूप में, हमने पढ़ा काउंटर करंट मैकेनिज्म, जहां पे कॉन्सेंट्रेशन या फिर डाइल्यूशन ऑफ यूरिन या फिर कुछ भी होता है तो वो वहां पे होता है हेनलेस लूप में. ठीक है, ट्रांसफर ऑफ यूरिन, ठीक है, विच इज डन बाय यूरेटर. यूरेटर ट्रांसफर करता है. स्टोरेज इज सिंपली डन बाय यूरिनरी ब्लैडर. ठीक है, तो बहुत इजी था, यूरिनरी ब्लैडर. नाउ लुक एट द ऑप्शंस हियर विच इज मैचिंग. A4, हाँ A4 दो में है तो C एंड D को हटा दो. देन B1 हिंडलेस लूप, हाँ तो A को हटा दो, B इज द करेक्ट ऑप्शन. C2D3. ओके, सो आंसर फॉर दिस क्वेश्चन इज ऑप्शन बी. बेटा ये बहुत सिंपल क्वेश्चन था, यू नीड नॉट टू वरी दैट मच रिगार्डिंग दिस. Okay, so that's all for today's class. Okay, we have solved enough questions. If you still have any doubts, you can contact the teachers available. Okay, so thank you for attending. Thank you so much. We'll meet soon. Okay, bye bye everyone. Bye.